We're going to take a look at capillaries and the lymphatic system. Remember that capillaries are the site of exchange between the blood and the tissue. Capillaries have a very small diameter, as you can see on this picture on the bottom. They are so narrow that the red blood cells actually have to bend and flex just to flow through them. So you would think that the capillaries would have a high resistance to blood flow. But because there are tons of capillaries in the body, there's over 25,000 miles of capillaries. If you add all of their cross sections together, you get a very large cross-sectional area for the capillaries, which makes them like a wide straw. And remember that if you're drinking through a wide straw, there's very little resistance to flow. So most of the capillaries are within about 1 millimeter of a capillary, since there are so many capillary beds in the body. And the capillary walls are very thin, they're just one cell layer thick, so things don't have to diffuse very far to get to the cells. And the other thing that makes capillaries really good at exchanging things with the tissues is that the blood flow through the capillaries is very slow. So that gives it time to drop things off and pick things up. Here's just a microscope view taking a look at a small artery here, and then we've got an arteriole branching off, and then you can see all of these capillary beds. So lots and lots of capillary beds, so cells are not going to be very far away from a capillary to do exchange. So these graphs are important. The top graph in green is just showing you the total cross-sectional area of the different blood vessels. And you can see that the capillaries, because we have so many capillaries, they have the largest cross-sectional area. So they're like a very wide straw, very little resistance to flow. And then the bottom graph in blue is looking at the speed of blood flow through the different blood vessels. So you can see that the speed of blood flow through the capillaries is very slow. And that's good because that gives you plenty of time to do exchange with the tissues. The reason that the blood flow is so slow through the capillaries is that it's kind of like the arterioles coming into the capillary beds. They're like a river, and then the capillary beds are going to be like a lake. So because the capillaries have such a large cross-sectional area, they're like a big lake. And as you know, water flowing into the lake will really slow down and will not flow very fast. That's what the capillaries are like. So they're like a lake. Capillary walls have small gaps between them, and that makes exchange between the blood and the tissue a lot easier because things can pass through those narrow gaps. There are a few areas in the body where the capillaries have much wider pores or fenestrations. The word fenestration means window, so it's just talking about a larger opening. So we'll find these capillaries with these fenestrations or pores in places like the kidneys, where we need to filter a large volume of fluid. Or they could be, for example, like in the bone marrow, where you're making the red blood cells, white blood cells, and platelets, and so you do need bigger openings to get the newly made cells from the bone marrow out into the blood. There are three ways to do exchange across the capillary walls. Most commonly, simple diffusion is used, but we can also use transcytosis, and in the brain, where we have the blood-brain barrier, we do use transport proteins. So here's just a picture of a capillary, and notice we've got the small gaps in the walls of the capillaries between the endothelial cells. And if you have something that is hydrophilic and small, so like ions, glucose, amino acids, they can just go right through those gaps and just do simple diffusion to move between the blood and the tissues. If you have things that are hydrophobic, so like oxygen and carbon dioxide, lipids, they will just go right through the endothelial cells. And then if you have proteins, for example, if you have maybe insulin hormone, that's a protein, and if you need to get it from the blood out into the tissue, you would do transcytosis to move that across. And that's because proteins are too big to fit through the gaps in the walls of the capillaries, so normally proteins are just going to stay in the blood. unless you move them across by transcytosis. In the brain, we have the blood-brain barrier. So remember the astrocytes, those glial cells, they will reach out their feet and stimulate the endothelial cells to form tight junctions between them. So there's no gaps in the walls of the capillaries in the brain. And so if you want to move something that's hydrophilic from the blood into the brain, you've got to use a transport protein. And so the blood-brain barrier is just there to help protect the brain, because the brain has to have very specific conditions in its environment to keep the neurons working well. As we move to our capillary beds at the arterial ends, we have the precapillary sphincters. They're just rings of smooth muscle that surround the capillaries, and they will contract and relax in response to local factors. So they'll be affected by oxygen levels, carbon dioxide levels, pH, things like that. So when the smooth muscle in the precapillary sphincters contracts, that will decrease blood flow into the capillary bed, and then that tissue will not get as much blood. And then when the precapillary sphincters relax, that'll increase the blood flow into the capillary bed. And here's just a picture just showing you the arterial, and then we've got the capillary bed here, and then on the other side we've got the venule taking blood back to the heart. And then notice these precapillary sphincters. So they will control the blood flowing into these capillary beds. Capillary exchange is important for two reasons. The first is that obviously it's necessary to exchange materials between the blood and the cells. And the second reason it's important is that it allows you to distribute the extracellular fluid between the different compartments. So remember, the extracellular fluid is made up of the plasma, which is the fluid part of the blood, and then the interstitial fluid. So if you're getting dehydrated, maybe you're sweating a lot, it's a hot day, and you're losing some of the plasma, that fluid, out through the sweat, you can actually replace some of that by moving some of the interstitial fluid into the capillaries and replenishing that plasma. So there's a couple of things that will govern how fluid is going to move across the capillary walls. So at the arterial end of the capillary beds, we're going to do filtration. So this is where the fluid part of the blood gets pushed out into the tissue. And then at the venule end of the capillary bed, we're going to absorb the fluid, or most of it, back into the venule to be taken back to the heart. We don't quite reabsorb all of the fluid that we filtered out. About 15% of the filtrate will be picked up by lymphatic vessels, and then those will eventually dump into the veins right near the right side of the heart. So there's two forces that affect filtration and absorption. So these are really important. I would star these. So blood pressure is going to favor filtration, so pushing the fluid from the blood out into the tissue. And then the osmotic pressure, which has to do with the proteins that are in the blood. Remember that water likes to follow the solute. So proteins are solute, and so water likes to follow the proteins that are in the blood. So that will favor absorption at the venule end. So the two forces for fluid flow across the capillary walls, we have the hydrostatic pressure, which is just the force due to fluid. So remember, hydro meaning water. And the main one there is the blood pressure that's in the capillary. And then the osmotic pressure is that the water likes to follow the solutes. So the solutes are the proteins, and so the proteins will pull the water back into the blood at the venule end. This picture of capillary exchange is really important, so make sure you star this and know this. So here's the arterial end of the capillary bed here, and then at the other end we've got the venule end of the capillary bed. And we've got a couple of forces that are affecting which way the fluids are going to be flowing. So at the arterial end, we've got the hydrostatic pressure of the capillary. So this is the blood pressure. The blood pressure at the arterial end is 38 millimeters of mercury, and that blood pressure is going to favor filtration. It favors pushing fluid out into the tissues. And then the hydrostatic pressure of the capillary at the venule end, the blood pressure drops to 16 millimeters of mercury. And that's because the further you travel, the more resistance you encounter, the more friction, and so that will decrease the blood pressure as you travel further. But again, the blood pressure, even though it is lower, it's still favoring filtration. You're still wanting to push fluid out into the tissues. The other place that we have fluid and another hydrostatic pressure is out in the interstitial fluid. So the hydrostatic pressure of the interstitial fluid is pretty low. It's only 1 millimeters of mercury. And that's because we don't have as much fluid out in the tissue as we do that's in the blood. But the fluid that is out here in the tissue, it would favor absorption and pushing fluid back into the capillary bed. And the hydrostatic pressure of the interstitial fluid is the same at the arterial and the venule ends. So to figure out our net hydrostatic pressure from the blood pressure and from the interstitial fluid, we're just going to subtract the two because they are opposing each other. So the net hydrostatic pressure at the arterial end is 38 minus 1, so it's 37. And then at the venule end, the net hydrostatic pressure is 16 minus 1, or 15. Okay, so that's the forces from the fluids, so the hydrostatic pressures, but the other pressure that affects which way fluid will move is the osmotic pressure. So this is due to the proteins. So remember that water likes to follow the proteins. And there are a lot of proteins in the blood, which we're going to see in chapter 15. So the osmotic pressure of the capillary from all these proteins that are in the blood is 25 millimeters of mercury. And that's going to favor absorption. So I've drawn a little picture over here, just kind of looking at an end view, looking inside the capillary. You can see lots of proteins in the blood. And just remembering that water likes to follow the solutes. So that osmotic pressure of the proteins in the blood, that's 25 millimeters of mercury. So that favors absorption, pulling the fluid back into the capillary. And the osmotic pressure of the capillary, the proteins in the blood, is the same at the arterial and the venule ends. And that's because the amount of proteins stays the same at both ends. Because remember, the proteins are too big to fit through the gaps in the capillary walls, so they just stay in the blood. And then the other osmotic pressure we have is out in the interstitial fluid. But basically there's very little or no proteins out in the interstitial fluid, so there's really not an osmotic pressure out there. So osmotic pressure of the interstitial fluid is 0 at both the arterial and the venule ends. But if we did have proteins out in the tissues, remember that water likes to follow the solutes, so that would favor filtration. So that would draw the water out of the blood if there was a bunch of proteins out in the tissues. So our total osmotic pressure coming from these proteins is just 25 minus 0, and that's the same at both ends. So our net osmotic pressure is just the osmotic pressure of the blood, which is 25. Okay, so now we need to compare the pressure from the fluids, so the hydrostatic pressure, and this is mostly due to the blood pressure. And then we're going to compare that to the osmotic pressure, the pressure from the proteins that are in the blood. So you can see that the blood pressure at 37 is bigger than the osmotic pressure from the proteins in the blood. And remember that the blood pressure favors filtration. So at the arterial end, we're going to favor filtration and push fluid out into the tissues. So that's good because we want to drop stuff off at the cells. And then at the venule end, you can see that our hydrostatic pressure, the blood pressure, has dropped, so the net pressure is now 15. And you can see the osmotic pressure of the blood from the proteins is at 25, so that's much bigger. And so that wins, and so we're going to favor absorption at the venule end. And that's good because after you've dropped off some of the fluid containing the glucose and ions and oxygen, whatever else, you can switch that around the cells out there, and then they can pick up things they need, get rid of waste products, and then we can reabsorb all that back at the venule and take that back to the heart. There is about 15% of the fluid that doesn't get reabsorbed, and so we're going to take that away in the lymphatic vessels, and then the lymphatic vessels eventually open up and dump that extra fluid into the subclavian vein, which is right near the right side of the heart. Okay, and then this is just a summary of what we just went over. And again, another summary. And just to answer our questions here, so looking at our osmotic pressure in the capillaries, so remember it's high in the blood, 25 millimeters of mercury, and it's high because there's lots of proteins in the blood. And remember, that's going to favor absorption. Water follows the solutes. It wants to be where the solutes are, so it's going to pull water into the capillary. The interstitial fluid, the osmotic pressure out there is pretty much zero, and it's low because there's no or very few proteins that are out in the interstitial fluid. So there's not a lot of proteins out there, and so water doesn't want to go out there, but if there were more proteins, it would favor filtration. And then this is just kind of a nice summary view. Just remembering that when blood comes into the capillary bed, at the arterial end, we're going to favor filtration, push some of the fluid, part of the blood out into the tissues, drop stuff off. And at the venule end, we're going to favor absorption and reabsorb that fluid and take that back to the heart. And then we do have a small amount of fluid that we didn't reabsorb that will get taken away by the lymphatic vessels, so they'll drain the extra fluid from the tissues. Okay, I will ask you about these different forces that act on the capillary walls. I'm not going to ask you these strange symbols. Okay, I'll just say the capillary hydrostatic pressure. And I would want you to know that that favors filtration. So just know like which direction it's wanting to force the fluid to go. So just make sure you know that for the exam. So if we upset any of these forces acting across the capillary wall, we'll get edema or swelling in the tissue because we have too much fluid that's remaining in the tissue. So let's just take a look at some examples of things that can cause edema. So what would happen if we increase capillary filtration? So what if we are pushing more fluid out at the arterial end into the tissues? So we're not going to be able to keep up with absorbing as much fluid, and so the fluid will build up in the tissue and we'll get edema. So this could happen if we increase the blood pressure in the capillaries. So for example, if you're standing on your feet all day, so by the end of the day your feet are sore, they're a little bit swollen, and that's because gravity will increase the blood pressure in the capillary beds in the feet, and that'll push more fluid out, more than you can reabsorb and drain away with the lymphatic vessels, and that'll cause edema. We also looked at edema being caused by heart failure. So remember, if the left side of the heart fails, the blood backs up into the lungs, and the blood pressure in the capillaries gets too high, and that pushes out too much fluid out into the lungs, and we get pulmonary edema. And then remember, if we talked about the right side of the heart failing, blood backs up into the vena cava, and then that will push fluid out into the abdomen, into the abdominal cavity, and that causes ascites, or out into the limbs, and that will cause like limb edema. And then there's a couple of other things that will increase pressure in the capillary beds, like maybe you're bedridden, blood's not flowing very well, it backs up, and then if your kidneys have shut down and you're not able to get rid of the extra fluid, you're not able to produce urine, that extra fluid will build up in the blood and increase the blood pressure as well. What if we increase the interstitial fluid osmotic pressure? So what if we increase the amount of proteins out in the tissues? So maybe you cut your skin and the blood is leaked out with all those proteins out into the tissue, that will then attract fluid from the blood out into the cut area, and that will cause swelling. And then if we decrease capillary absorption, so how we do that is if we decrease the capillary osmotic pressure. So what if we decrease the amount of proteins in the blood? So that's called hypoproteinemia. There's a lot of different causes for that, but maybe you have liver disease and you're not making as many proteins in the blood anymore, or maybe you have kidney disease or GI disease and you're losing a lot of proteins through those organs. And then the last one is, what if you block the lymphatic vessel? So maybe you have cancer and you have a tumor that's blocking that lymphatic vessel. You can't drain that extra fluid and that'll build up and cause edema as well. So these are just some things just to kind of think about, because you will be seeing these in patients. And if I give you a scenario, you could kind of figure out, what would happen. So you don't need to memorize all these different causes, but just kind of think about, if we mess up some of these pressures, that can cause edema. Okay, and the last thing to talk about is the lymphatic system. So we're going to talk about this again when we get to the digestive system and immune system. So we've got these lymphatic vessels that are going to be used to drain the extra fluid from the tissues, and then they will dump that back into the blood. The lymphatic system is also used by the immune system. A lot of our white blood cells will travel through the lymphatic vessels and hang out in places like the lymph nodes and spleen, tonsils, things like that. And then our digestive system will actually move the fats that you've eaten from your diet. Remember, we package those into those big giant chylomicrons, those lipoproteins that are too big to fit in the capillaries. And so we put them into the lymphatic vessels and transport them that way, and then dump them into the blood. So here's just a picture of the lymphatic system, just showing you some lymphatic vessels draining the extra fluid from the tissues, and then that will get dumped into the blood eventually near the right side of the heart.